Hello, my name is Angela Roca and I work as a project manager in the R&D department at Provital. Today I'm going to talk about our paper that focuses on bitter taste receptors, the one detecting bitter substances such as cocoa, coffee or beer. During the last decades, there have been several scientific papers from different authors reporting that the skin can smell, taste and see. Epidermal keratinocytes respond to visible light, to different odors, and even have a bitter taste. But what is the role of these receptors in the skin? We will come back to the skin later. First, let me tell you what we know about the bitter taste receptors. Obviously, they have been extensively studied in their original tissue, the taste buds of the tongue. We know there are 25 different bitter taste receptors capable of detecting different bitter chemicals and plant-based molecules with a diverse chemical structure. These receptors initiate the bitter taste perception in the oral cavity and work as a warning signal against the ingestion of potential toxins. However, growing evidence indicates that bitter taste receptors are widely expressed throughout the body, in tissues such as the nasal epithelium, brain, heart, gut and lungs, where they mediate diverse non-tasting functions. Several research groups have characterized the function of these extraoral receptors, revealing that their physiological roles are related to toxin metabolism, toxin elimination, defense mechanisms, and protection against bacteria infection. But what about the skin? As I mentioned earlier, bitter taste receptors have been identified in the skin, particularly in the epidermis, where they participate in the keratinocyte differentiation as well as a role in immunomodulation. Until now, these receptors have not been investigated in other dermal cells. Nature is a rich source of medicinal plants, and many plant compounds have bitter taste to protect the plant against external aggressions. So we can think that as the skin acts as a barrier, the presence of bitter taste receptors could be a mechanism to protect the skin from the environment. And if we consider the different functions bitter taste receptors exert in these extraoral tissues, it was logical for us to think that bitter taste receptors, when activated, could trigger an immune response and contribute to enhancing defense mechanisms of the skin. Thus, we have demonstrated for the first time the presence of bitter taste receptors in dermal fibroblasts and how they are responsive to natural bitter compounds present in a hop flower extract. Indeed, this activation of the receptors boosts the cellular mechanisms to reduce the inflammation and enhance the defenses to protect the skin from the external aggressions, improving thus the cutaneous comfort in reactive and allergic skin.